Assalamualaikum. My name is Nur Izzati binti Zainuddin, and for today's presentation, I'll be presenting you my final year project entitled Sequencing Batch Integrated Fixed Film Activated Sludge Membrane Process for Treatment of Tapioca Processing Wastewater. As for today's highlights, I'll be covering 4 chapters, which are Introduction, Methodology, Results and Discussion, and lastly Conclusion and Recommendation. And each of these chapter will have a few sections. Firstly, I'm going to introduce small-scale tapioca processing industry, where the tapioca flour is actually originated from the cassava plant. And this cassava plant is commercially or widely used in pharmaceutical, textile, and also cosmetic. Besides, the tapioca processing industry is also popular in Asian countries like Malaysia, Indonesia, and Thailand, where they practice this industry in small scale. Okay, as you can see here. Since it is small scale, the whole process is not fully automatized yet, whereas some part of it still requires human energy, for instance, to do the peeling. Next, the production processes are usually in semi-batch wise, hence the name just now, and it is being done over a 1-day cycle. After that, the tapioca wastewater will also be discharged intermittently, and usually this discharge happens in the morning session. Then since the small-scale tapioca processing industries are scattered in places, it is hard to implement an efficient wastewater treatment. Thus, the tapioca wastewater discharge will give adverse effect or tension to the environment as it contains high organic pollution or nitrogen and also phosphorus. Thus, the chemical oxygen demand discharge is from 9,000 up to 13,000. And because of this reason, it is introduced an anaerobic treatment to remove the COD, where it is called as Upflow Anaerobic Sludge Blanket Reactor UASB. And it is proven that this system has the ability to remove COD from 92% up to 93%. Somehow, this reactor is also deemed to be complicated. This is because this reactor actually cannot withstand with high level of suspended solid present in the tapioca processing wastewater. And to use this reactor, the suspended solids need to be lessened first, and it can be done by using the anaerobic filter, or we call it as AF. Okay, and then that's why the whole treatment is actually not user friendly, and it will cost the business owners a lot, as it needs 2 kind of reactors to proceed with the COD removal. Because of this reason, okay, I will introduce a system called Sequencing Batch Integrated Fixed Film Activated Sludge Membrane Process, or we call it as FYP2, where this system is actually implementing a gravity-driven membrane coupled with biofilm. What is gravity-driven membrane? Gravity-driven membrane is actually basically a filtration done without using a power pump, like how membrane bioreactor uses normally. Okay, it utilizes the gravity, and also the biofilm. Biofilm is a clump of bacteria where it is adhered together, and it needs organic nutrients to survive. So, in other words, the biofilm will be used as a secondary filtration, where it will do the biodegradation part, where it can utilize the organic present in the tapioca processing wastewater. So it acts like a secondary filtration, a part of the membrane bioreactor. Okay, where this membrane is actually is called as polyacrylonitrile membrane or PAN membrane. So, as our hypothesis, SBI-FZM could help to make the existing treatment to become less complex, more convenient, and also more economic. As I have mentioned just now, that the UASB is actually complicated because it needs to have a pre-treatment before using it due to the disability where it cannot withstand with high suspended solid present. And then this SBI-FZM is also deemed to be a new exploration of technology, as there is no research done yet regarding this technology in treating the wastewater treatment of tapioca processing industry. Okay, so our problem statement is first, due to the nature of tapioca processing industries in Asian countries where they are practiced in small scale and also scattered in places, it is hard to implement an established treatment system where it is considered to be untenable or not viable due to the mentioned issues just now. Okay, and then the membrane process, such as in membrane bioreactor, okay, it usually experiences fouling, and this causes them problem because the fouling needs to be cleaned, whether chemically or mechanically disinfection, and it becomes a bit complicated for the business owners to operate. So that's why gravity-driven system has been introduced and applied as stand-alone process, the SBI FSM just now. After that, for our objective, okay, there are three objectives to be presented in this FYP. First, to evaluate the impacts of hydrostatic pressure on membrane permeability for clean water and activated sludge gravity-driven filtration, where we will implement three different pressures, which are 2.2, 3.2, and 10 kPa. Okay, and then we also will investigate the impact of sludge concentration effects on permeability of membrane filtration, where three different concentrations are measured from 100% concentration, 50 to 25 concentration of activated sludge. And lastly, we are going to assess a non-steady state operation of SBI FSM in long-term operation, where it actually operates in sequential batch, and we are going to focus particularly on the biofilm contribution to remove the organic, and we're also going to focus on the hydraulic performance. Next, as for the scope of study, the SBI FSM system was operated using lab scale and proven to achieve a good efficiency of COD removal by utilizing the biofilm, pen membrane, and also an aerator. Okay, so this aerator was actually meant to supply the oxygen for the biofilm, as biofilm cannot survive without two things, which are organic nutrients and also oxygen supply. Then, the effect of hydrostatic pressure on the permeability of clean water and activated sludge was investigated at steady heads of 0.22, 0.32, and 1 meter, corresponding to the pressure of 2.2, 3.2, and 10 kPa accordingly. After that, the feed and permeate samples were recorded daily for COD and total solid analysis, as these analyses were intended to monitor the biofilm behavior. In addition, the literature review, okay, I present it in a table form, where this table actually demonstrates the findings of biofilm by the researchers. And as you can see here, the source of the feed water varies. Okay, the most common one is river water. Okay, and then next they also test with the pond water, diluted wastewater, grey water, synthetic grey water, rainwater, and lastly seawater. Where for my research, I use synthetic tapioca processing wastewater as my source for the feed water. Okay, and then the applied pressure for each of these experiments also varies. Okay, where the smallest one is 0.03 bar, which equal to 3 kPa, and the largest one is 0.615 bar, which is actually 61 kPa. Where from my research, the smallest one is 0.02 bar and the largest one is 0.1 bar. So since the source of the feed water and applied pressure are different, that's why the stable flux for each of these experiments also different from one another, and it equals to the different stable permeability. And as you can see here, okay, the used membrane here is also different regarding the type of the membrane. And the most common one is polyether sulfone, where from my research, I use polyacrylonitrile or the pen membrane just now. Okay, and as I have mentioned earlier in my presentation, the biofilm is actually a clump of bacteria, and the clump of bacteria is possible due to the extraction of the extracellular polymeric substance. For this research, we call it as EPS, because this EPS will trigger will prompt the bacteria to interact to one another, and soon they will be adhered together on the solid medium, where for my research, the solid medium is actually the pen membrane. So the clump of bacteria will be adhered on the pen membrane surface. Okay, and the EPS substance also act as metabolism, and it is made up from carbohydrate and also protein. Okay, and as for the methodology, first the sludge cultivation was done for a week. Okay, and the membrane cleaning was performed using a detergent solution, and it was soaked in a solution for a day. After that, 3 membrane filtration setup was done, and an aerator was equipped with this setup. And then the activated sludge was also fed with 2 grams of urea, 2 grams of glucose, 3 grams of potassium dihydrogen sulfate, and 30 grams of tapioca flour. Okay, and then after the cultivation of sludge was done, the short filtration was performed for an hour with 10 minutes of time interval by using the clean water and activated sludge as the feed water. Okay, and then it was proceeded with the semi-batch long filtration, where this semi-batch long filtration was done for 43 days and recorded. And it was also done by using the activated sludge, and we utilized the reactor A for this filtration. Okay, after that, sampling and data analysis of permeate, activated sludge for COD, and MLSS analysis. MLSS analysis is for total solid analysis. Okay, and then lastly, submission of the final year project 2 in week 9. Okay, and this is for the materials. Okay, where this is the activated sludge. Just now we cultivated and we fed with some ingredients. Okay, and then this is the pen membrane. This is the aerator. Okay, and for the experimental parameters, we have two, which is short filtration and also long filtration. Okay, and then these are the setup illustration for the reactor A, reactor B, and reactor C. Even though you cannot see clearly for the reactor C, but reactor C is almost the same, except that the height is 1 meter, which is the longest among the 3 reactors. Okay, and then this is the calculation. This is the formula for the calculation of the permeate flux and also permeability. Next, for result and discussion, for short filtration part, the effect of hydrostatic pressure on the clean water and activated sludge filtration is illustrated by the figure 12 here. Okay, and then figure 12 also shows that at low transmembrane pressure, the hydrostatic pressure strongly affects the filtration of clean water and activated sludge, which explains why every reactor here for these two parts give different permeability readings, when generally it is agreed that the permeability readings should be constant at any applied pressure when the membrane reach permanent compaction. Somehow, due to the significantly lower applied pressure, the membrane compaction phenomenon seems to be reversible. Okay, where it is also observed that the pen membrane actually constricts when it is under pressure and expands back when the pressure is released, which explains why the permeability decreases when the applied pressure increases. Okay. Meanwhile, for the activated sludge part, the permeability readings are much lower due to the cake layer fouling phenomenon, where the cake layer is instantly built at the initial stage, okay, driven by the convective flow of the permeate. And then next, for the short filtration, regarding the effect of concentration on the permeability, figure 13 shows that there are 3 different concentration types, okay, where the highest one is C3, followed by C2 and C1. And as expected, the highest concentration will have the lowest permeability readings. Okay, somehow comparing this figure 13 with figure 12, okay, the decrement is only slightly, okay, and this is due to the flux consolidation in the cake layer that enhances the cake compactness, where high sludge concentration is more profound. For the long filtration part, the filtration performance is analyzed and illustrated in the figure 14, as you can see here. For the first 7 days, the permeability readings are considered to be stable even though there is no cleaning done. But then starts on day 7, it starts to drop. This is because the formation of biofilm starts to happen after day 7. And starting from here, it keeps dropping throughout the filtration performance. Okay, this is because the formation of biofilm starts to get thicker and denser throughout the time, so the hydraulic resistance increases, which explains why the permeability decreases over time. And as you can see here, generally, the permeability readings are much lower compared to the conventional membrane bioreactor permeability readings. This is because of there is no cleaning done, and if you wish to restore the reading, you can perform the chemical or mechanical disinfection. And the permeability readings are also influenced by the applied pressure and also the nature of the feed. Nonetheless, due to the absence of membrane membrane fouling control, the SB-IFASM operation turns to be very simple with minimum energy input. For the total activated sludge mixing, as you can see here in figure 15, for the first phase here, for the feeding one part, okay, observe this part first, there is a declining trend. Okay, this declining trend actually indicates the importance of mixing in SB-IFASM system. For the first 8 days, it's stable, the declining is stable, but then the declining starts to be dramatic starting from here until here. And this is because of the partial sludge settling and also sludge bulking. And this can be illustrated with this picture here. And this forming actually resulted from the instant feeding where the organic content will rise dramatically. So starting from here, okay, the feeding style is changed and then it increases again on day 23. But then after day 33 or 34, it starts to drop again and the trend is exactly the same like this. Okay, but then this is due to the formation of biofilm where it gets thicker and denser, so it needs more food to sustain. So in other words, for the feeding 2, okay, feeding 2 should be more compared to the feeding 1 to provide energy for the biofilm to do the degradation. And then the total solid analysis is also vital to check the current biomass level of the sludge so the bacteria can be sustained throughout the experiment. And is, as you can notice here, okay, from the figure 15 with the figure 14, one can actually expect the logic where if the total solid decreases, the permeability should increase, but it happens otherwise for this part. Okay, this is due to the biofilm biological system. So in other words, it is important to maintain the biological system, okay, for the biofilm so that the result will be like what we have expected earlier. Okay, and for this part, okay, where figure 16 illustrates the chemical oxygen demand removal. Okay, as you can see here, generally the COD removal consistently high, which is more than 90%, and this is thanks to the biofilm utilization. And as you can see here, since it drops, okay, but then since before this we changed the way we feed the biofilm, the COD removal starts to increase again, which actually illustrates that the importance of feeding for the COD removal. Okay, and then as a conclusion, okay, our conclusion should actually answer the objective. Okay, so for the impact of hydrostatic pressure, the result of short-term filtration tests for clean water and activated sludge filtration show the permanent impact of pressure in compacting the membrane, thus lowering the permeability. Okay, and then for the effect of sludge concentration, the lowest concentration of sludge give the highest reading of permeability as expected. And lastly, for the organic removal, the contribution of biofilm in enhancing biological performance is demonstrated by constantly high of COD removal, which is more than 90%, making it attractive and more economic for the application in small-scale tapioca processing industries. As a recommendation, the biofilm should be analyzed using software like MATLAB to perform the calculations and analysis. Okay, and then the accuracy of the results can be enhanced by analyzing the nitrogen and phosphorus for biofilm performance. And lastly, the sludge can also be improved if the sludge is fed automatically and equipped with a proper mixer to mix the wastewater evenly. So that's all from me. Thank you.